Good evening, and welcome to Winchester Academy's program this evening. My name is Maggie Jones, and I am a trustee on the Winchester Academy Board of Trustees, and I am also chair of the program committee. Thank you to the City of Waupaca and to our producer, Josh Warner, for providing the technology to bring this program to you in all the multiple formats that we use. And I also want to say thank you to the Granite Cellar Bar over at the Danes Hall here in Waupaca for hosting a viewing party tonight. So those of you who are at the Danes Hall watching us, hello. Our next program will be 1 week from tonight on October 25th at 6:30 p.m. And we will be hosting Gregory Aldretti. His talk is History or Hollywood? Ben-Hur and Gladiator. Tonight's program is sponsored by Rita and Bob Danielson. We especially appreciate their financial support, and we're also so grateful to them because they were the ones who recommended our speaker to us tonight.
Thank you.
Following the program, we will have a Q&A session, and the questions can be submitted via Facebook Live or by telephone. And if you call in your question, the telephone number is 715-942-9917. Again, that phone number, 715-942-9917. And our speaker tonight is Dr. David Gamm. Dr. Gamm earned his MD and his PhD degrees at the University of Michigan at Ann Arbor. He currently serves as Associate Professor of Ophthalmology and Visual Sciences. He is the director of the McPherson Eye Research Institute and holds the Sandra Lemke-Trout Chair in Eye Research at UW-Madison. Dr. Gamm is both a clinician and a researcher. His clinical practice is in pediatric ophthalmology, and his research interests focus on human retinal development and disease. And David is joined this evening by his colleague, Michael Gamm, who works in development at the McPherson Eye Research Institute. So please join me in welcoming our speaker, Dr. David Gamm.
Thank you very much. It's very, very kind, and I really appreciate being able to be here and to speak to you. And I do encourage folks to call in later. I usually do the Larry Mueller show at Wisconsin Public Radio once a year, but this year I didn't do it because of COVID, so I kind of miss it. So I do encourage people to do that. So as mentioned, I'm a physician scientist, which means I see patients. I do surgery at the University of Wisconsin, predominantly with kids, but also I see some adults as well. And I was mentioning earlier that I like that because most of the time I can help those patients, whereas in my laboratory we really concentrate on things, diseases that for right now have no cures, that have no treatments. So the clinics are an outlet for me where I can actually help folks, and then the research is what I concentrate on to develop new therapies for folks that really have no place to turn right now. And that's really the topic that I'm going to talk about today is where does stem cell technology fit into all of that? Stem cells is something that's thrown around all the time now, and it's used more often than not to try to fool people and to get them to open up their pocketbooks and do things that may be harmful not only to their finances, but maybe also to their health or their vision. So I'm going to try and separate the hope from the hype for you today, and but to start with that, the hard part about trying to piece apart things that's used for ill-gotten gains, like stem cell therapy sometimes are, is that it takes a little bit of education to understand exactly what is a stem cell, what can it be used for, because there are limitations to it, and how would you apply it. And of course, there's different systems that you could apply it to: brain, for joints, liver, things like that. But of course, my interest is in the eye. So I'm going to talk about stem cell biology and how it applies to the study of the human eye, and ultimately to the development of treatments to try to prevent disease or to restore vision once folks have lost a good amount of their vision to start with. So I always start with this idea about, stem cells invoke a lot of different feelings in folks. There's certainly ethical issues with some types of stem cells, not all, and we'll talk about the different types. There's also political aspects to it, and then there's medicine, and then there's science. And I'm going to concentrate in my talk on the medicine and science portions of it, but I'm perfectly happy to talk to folks about their other interests that are also very important and worth public discussion. So a couple disclosures. I do have my laboratory, and my work over the last 10, 15 years has developed a number of patents that are all patented through the University of Wisconsin on a lot of the technology that I'll be talking about today. And there is a company in Madison, Opsis Therapeutics, that was a spin-off of this technology through the university, through WARF. And then that is, and its mission is to take the technology, the stem cell work that we do, and to bring it to patients, both for childhood blinding disorders as well as adult onset macular degeneration. So I need to disclose that as well. And then this is the outline for the talk today. I'm going to start with just giving you a brief kind of 101 primer on cells. What are cells in general? And then work into what are the cells of the eye, what is the retina, so some anatomy. And then use that as a foundation to talk about different diseases of the eye that ultimately we hope stem cells may have a treatment for. And then lastly, talk about stem cells in general. And then with that information, hopefully we'll be armed with enough knowledge so that I can then talk about how we're going to use the technology that we've developed and other people have developed around the world to hopefully develop these treatments that we're looking to create. And then I'll, of course, acknowledge the people who do the real work in my lab. Okay, so our bodies are made up of trillions of different cells. Each one of those cells is like a factory within it, in and of itself. And if you think about drugs and gene therapies and stem cell therapies, they all act in fundamentally different ways. And we hope that all the cells in our bodies stay healthy for our whole life, but oftentimes they become diseased or dysfunctional. And fortunately, most of these diseases that we get, there's a drug or there's some sort of therapeutic that can act on that disease cell to turn the tides and make it healthy again. But unfortunately, with degenerative diseases and specifically, so if you think about degenerative diseases, what makes them different than other types? And the problem with those is that they end up disintegrating. So the diseases that you have, if they continue to progress, will lead to the destruction of that cell type. And at that point, there really is no way to put that cell back together again. Okay? So that's not too bad in cases or in tissues or in organs in your body that have the ability to regenerate. And a number of them do. So, and those are the tissues and cell types that undergo natural degeneration over time. The skin cells on the surface of your skin that line your gut, your blood cells, they all have a finite lifespan, and over time they need to be replaced. And so your body's set up to do that. So they'll have these, what we call tissue-specific stem cells. They're in your bone marrow, they're deep in your skin, and their job is to continuously throughout their life to divide and to repopulate those cells that are going to get lost, because they're supposed to get lost over time. However, and like I said, this is blood cells, skin cells, so there's different places in your body that can do this. And there are diseases that prevent you from doing this, and then like aplastic anemia, and they can be devastating. But if you're otherwise healthy, there's not a problem. So unfortunately, though, a lot of places in your body can't do this naturally. And the prominent ones that we think about are in the central nervous system. So brain, spinal cord, and the retina, which lines the back of your eye, we'll talk about a little bit later, that's also part of the brain. So it's part of the central nervous system. And what's common amongst all the cells in the nervous system is that they can't regenerate themselves once they've been lost. So that gives you the who's who of what you don't want to get in your life. So your Parkinson's, your Lou Gehrig's disease, your Alzheimer's, your retinal degenerative diseases, paralysis from spinal cord injuries, all those things cannot regenerate themselves. So once they're gone, they're gone. And so that's why I include here this little cutout of the eye with the retina in the back. So heart can't do it. The insulin secreting cells of your pancreas can't do it. Once they're gone, they're gone. That's why you get diabetes. And cartilage can't do that. So you have all the cartilage you're going to get in your life. So once it's gone, you have to replace it with some other means. And also the cells in the eye. So this is just an eye. And so if you look inside of it as ophthalmologists, when we dilate your pupil and take a look inside, this is what a normal eye looks like. The white disc is your optic nerve. That's the coalescence of 1.2 million wires that take all the information, the visual information from your retina, coalesce that into a cable that goes back into your brain, and then your brain does the seeing. All of the orange stuff you see here really is the blood that's behind the retina, because the retina is transparent. It's like cellophane. And those little blood vessels you see are feeding the inner parts of the retina, but the orange part is like a waterfall of blood that's feeding all of the rods and cones and photoreceptors that are clicking on and off all the time, detecting light. So it's really a grand central station of activity. And pound for pound, it's the most metabolically active tissue in your entire body, and the most electrically active tissue in your entire body. So it has a lot to do all of the time. So sometimes I think it's not a wonder that we have degenerative diseases of the retina, but rather, how do we live 100 years and have something that works, a factory that works this hard for this often, all the time, during the day and night, and how does it continue to work efficiently for that long of a period of time? So it's really a remarkable system, but unfortunately it doesn't stay remarkable for some people for their entire lives. So if a retina is working well, you get a nice visual field like this, these two boys with balls. It's also important to realize that when I talk about vision and blinding disorders, that can occur anywhere from the front part of the eye all the way to the back of the brain. So as I mentioned before, you actually see with your brain. So there are people who get head injuries. Their eyes are perfectly fine. Their cables, the optic nerves, are perfectly fine. But they get a devastating head injury or a stroke, and they're completely blind because they take out that primary visual cortex. You also can have, people probably notice, you could get a chemical burn and your cornea becomes opaque. You can't see then either. So there's a lot of different ways throughout this entire system, from the front of the eye all the way to the back of the brain, where you can go blind. My lab and people at the McPherson Eye Research Institute at the University of Wisconsin work on blinding disorders in that entire pathway. My lab in particular works on the retina, which is the thin film that lines the back of the eye. So I can take calls later on folks that have interests in glaucoma or head injuries, things like that, because I deal with those in my clinical practice. But my laboratory specifically works on the retina. So let's concentrate on the retina then. So you take that same eye, let's cut it in half, and look at it from the side. And so this is a schematic of your eyeball with the cornea being shown on the left. That's where light would enter through the pupil. The lens is that oval disc towards the left side of the picture. And then the orange area there is kind of a half sphere of the retina. And the inside part of that is filled with a jelly called vitreous. And so it's an optically clear jelly. We could talk about that again later too. All these little parts are fascinating. But I have to get to the gist of the talk, or you guys are going to be here for 2 hours. So the little white square there is what I'm going to blow up now. So if you take a little piece of the retina, I said before it looks like cellophane. And if you just take it out of an eye, it looks like a kind of a thick piece of cellophane, not too much to look at. But it's actually a very elegant layer cake of cells. So if you look at it under a microscope, it's not just an amorphous thick piece of tissue. It has specific cell types that are in a very precise order, in a very layered order. And so deepest within the retina are what are called photoreceptor cells, or the rods and the cones. These are the cells whose responsibility is to detect light. So they're the apex of your entire visual system. They're what starts the whole process. And the rods are usually more peripheral, and they give you your night vision, kind of your black and white vision. They're not really what we use under lighting like this. So under lighting like this, all of your rods are shut down because they're really out for you to walk around at night. And so your rods are shut down, and your cones, your red, green, and blue cones, are the ones that are working and giving you your fine vision, your color vision, and everything really that we prize as human beings, our ability to read, to recognize faces, things like to drive, things like that. And beneath that, so photoreceptors are super metabolically active, and they can't sustain that activity by themselves. So they have a helper cell, they have a handmaiden called the retinal pigment epithelium, which is a thin layer, a single layer of cells that's actually pigmented. It looks dark brown, and it supplies all the nutrients through that waterfall of blood behind it, all the nutrients to the photoreceptors, and takes away all the waste products. So this is kind of the Batman and Robin of vision. So the photoreceptors get all the credit, they get all the hype, and the retinal pigment epithelial cells do a lot of the work and never get any credit. But without the retinal pigment epithelial cells, the photoreceptors aren't long for this world. They can't do it by themselves. And then the photoreceptors, it's a very simple kind of electrical circuit. They're connected to what's called a bipolar cell that's in the middle of this cake. And those bipolar cells are connected to ganglion cells who produce the 1.2 million little wires that coalesce into the optic nerve. Right? So all of this is like a funnel that goes to the optic nerve and back to the brain. But it starts with the photoreceptor cells and the RPE cells helping them. So light passes through the entire thing. That's why it has to be transparent, because the photoreceptors are on the base of this. So if it wasn't opaque, the light wouldn't be able to pass all the way through and stimulate the photoreceptors. And then that signal goes all the way back through the system and back to the brain. And you have just trillions of these bits of information that are being processed by the brain every second. So I'm going to concentrate. So you say, OK, well, you're really focusing in on things, Gamm. So not only am I not talking about the entire visual system, I'm not even going to really talk about the entire retina. I'm going to talk about that Batman and Robin, the photoreceptors and the retinal pigment epithelium. Because a lot of what we deal with as we age, or also kids who go blind, has to do with dysfunction in one or both of these cell types. This is what the retinal pigment epithelium looks like. So right here, it's just kind of, I show it on a cross section on its side, so it just looks like a single layer. But if you peel the retina off and look at it from the top, it looks like these dark brown, blackish cobblestones. And so you have to imagine all the photoreceptors kind of sitting, poking into it from the air into the screen. And I'll try to show this here in a three-dimensional slide here in a moment. The photoreceptors, I showed you a cartoon earlier. There's something I drew. But this is what they actually look like. And they're very, I mean, I'm a little bit biased, but I think they're the coolest cell in the entire body. They have a cell body to them. They have a power factory that's chock full of mitochondria, so it can do all the metabolism I talked about before. And then it has this little antenna, this little proboscis that sticks down into the retinal pigment epithelium. And these are antennae that are filled with millions of these proteins that detect light. And it's specifically set up just to be an antenna to collect light. And collecting light is not a simple business. There are hundreds of different proteins and different connections that have to be made, and they have to work together absolutely perfectly in order for that signal, that photon of light, to be converted into a chemical signal and passed back through the system to the brain. And so any one of these little parts along the way, any of these little gears and cogs that don't do their job correctly, can throw a monkey wrench in the whole system, and that results in blindness. So again, it's really remarkable that this finely tuned watch works so fast for so long without going wrong in most people. So this is, there's no quiz on this, there's no exam. This is just to kind of give you a good sense of what all goes into the process of just that one cell detecting one photon of light. So getting back to the 3D, so hopefully you can get kind of a better sense of how these cells interact. So if you're looking at the retinal pigment epithelium, the photoreceptors kind of line up like grass. If you think of RPE like the dirt, and then the photoreceptors as the grass on the dirt. You kind of line up like that. And you got to think of that lining the entire back of the eye, and that's just the outer third of the retina. I'm ignoring all the other stuff that it's connected to. Okay, so that's when everything works right. So what about retinal degenerative diseases? What are the main types that we're concerned with, that we work on in the lab and in the Eye Institute, that really people have no place to turn for treatments, but that we want to fill that gap? So one large group, there's a lot of them actually, there's hundreds, but I'm going to concentrate on two kind of large categories of diseases. And then I can talk more about specific other ones if people have questions. The first one is called, the term is called retinitis pigmentosa. So this is over 200 different diseases. They're all inherited, so it's genetically transmitted. They're all very rare by themselves, and collectively they affect maybe 2 to 3 million people worldwide, we think. The problem is they affect kids, and so they have a really devastating effect throughout people's entire lives, and they affect entire families. And as a pediatric ophthalmologist, this is near and dear to my heart, and one of the things that really brought me into the stem cell field. So again, about 2 million people worldwide, affects younger individuals all the way from newborn infants to teenagers, kind of late single digits is when you start to show the symptoms. And then usually these folks by their early 20s are pretty profoundly visually impaired or blind. And there are a couple of therapies that maybe slow things down for 1 or 2 of the 200, but for the most part, there's nothing that we can do for them right now. And then the other large group is age-related macular degeneration, and I think everybody's heard that. It's something that scares a lot of folks. It's becoming a bigger public health problem. It's one of the largest, actually the largest, degenerative disease of the eye that we deal with in the back of the eye, in the retina, the outer retina at least. And it's growing because the population is aging as well. And people are living not only longer, but they're doing more, and they're more productive, and they want to do more things. So the loss of your central vision becomes more impactful than, say, it was 20, 30, 40 years ago. And AMD, as opposed to retinitis pigmentosa, is not rare. There's over 10 million people in the US alone that are affected by it. About 1 million of them profoundly, where they're legally blind or worse. And it's rapidly growing, as I mentioned before. And there are treatments for certain types. So you might hear about the wet form, where you have this bleeding. I'm going to talk about that a little bit. But even that, so what happens with the wet form is that you can slowly start losing vision, and all of a sudden you have this bleed. And it's possible now with certain medications, these anti-VEGF therapies, where you can dry up that bleed and people will regain vision. But that's superimposed upon this slow sort of forest fire called dry AMD. And this is a slow degenerative process that really has no treatments whatsoever. And so even if you could treat the bleeding portion of it, you're left with the underlying dry AMD that we can't do much about. And I'll get into more details on that. So going back to my diagram I used before, so in retinitis pigmentosa, the genetic defect is usually in the photoreceptors. So there's a problem with one of those cogs, one of those gears in the photoreceptors that causes it to deteriorate over time, and that throws the whole thing out of whack, and then they frankly die over time. It's also possible that there's a genetic defect in that helper cell, the retinal pigment epithelium. And as I mentioned, without that, the photoreceptors die thereafter. So the problem isn't with the photoreceptors, but if their helper goes away, the photoreceptors go soon after that. And this is what a child with retinitis pigmentosa might look like compared to what I showed you before, which is a normal healthy retina. They'll have these black areas where there's areas of degeneration. And you can see that the blood vessels are really thin and whispery. The optic nerve also changes as well. And a child who has a retina that looks like this would see something like this initially, probably in their late single digits, early double digits, but unfortunately, over time, they lose that central vision as well. So it's quite devastating. In age-related macular degeneration, the problem isn't in the photoreceptors. Actually, your photoreceptors are totally fine. The problem is in the retinal pigment epithelium. You get the initial death is in the RPE. Let me do that one more time here. And then slowly thereafter, is that the photoreceptors die in the absence of the RPE. In the wet form, this is the wet form, when the RPE dies, the blood vessels that are right behind it have no barrier. And so those blood vessels will grow up into the space that the RPE used to be, and then that will cause rapid death of the photoreceptors. So if you can get to them before that rapid cell death and get rid of those blood vessels, you can regain some vision. But again, that slow train that's going towards degeneration will always continue to inexorably move on. And this is what this looks like as opposed to a normal, healthy-looking retina. So the dry form, you can see this area where it looks like a cookie cutter. Somebody just kind of cut out the retina. And all of that area that you see is the retinal pigment epithelium and the photoreceptors that are no longer there. It's like a desert. And as you might imagine, that leaves a big scotoma, or blind spot, right in the center. The wet form has the blood that you see there. And if that blood were to go away, so you could treat that blood, you can imagine that's really big, right? You have this huge blind spot there. But even if you get rid of that wet blind spot, you're left with what's on the left. Okay, so that's the part that has no treatment currently. And so this is what somebody with a severe case of age-related macular degeneration would see, which is that center portion. So it's kind of an inverse retinitis pigmentosa. So what can you do? So I'm from Michigan originally, so I like automotive analogies. So if you think about, you use a car as an example. And if your car doesn't run because you ran out of oil, you can always put more oil in. That's like a drug, right? So the pistons are fine, the engine runs fine, you just ran out of something, you can replace that something. If you have, if the pistons then break, then you need to replace a piston. That's like gene therapy, where you have to replace a part. Cell therapy is like replacing the entire car. So there's no longer one part that's missing. The entire cell is no longer there. And that's the problem with all these degenerative diseases, is that slowly but surely the entire cell goes away. And so you're left with two options when you have no more cells left. You can either bypass that cell, so you can try and get something downstream to do the job of the cell that just died, or you can try to replace the cell that died. The bypass route is something called, in the retina, something called optogenetics, where they try to take those bipolar cells and those ganglion cells, the ones that are further downstream of the photoreceptors, and they try to get them to detect light. And that actually works, but it's very, very crude. So you can, light on, light off, some movement, something like that. So that is a promising way to go. It's not what we concentrate on. We concentrate on the replacement aspect of therapies. So, but that is where, when I started as a fellow and a resident at the University of Wisconsin, there really was not anything. There were no spare parts. So if you're looking to replace a cell type that's been lost, where are you going to get it from? And so at that time, late 90s or mid to late 90s, people thought, well, what if we had a living donor? What if we took it from somebody else's eye or the other eye? Problem is you blind that eye if you do it. So unlike taking a piece of your liver or a kidney where you have two of them, if you start messing around with a person's retina, you're going to blind them in that particular eye. So taking a healthy eye and blinding it for an eye that's unhealthy doesn't seem like a very smart thing to do. What about cadavers? What about deceased people that donate things? People talk about donating your eye. Well, when you do that, you're actually donating the front part of the eye, the window or the cornea. You can't donate the retina. And people actually looked at that in the 70s and 80s. Could you take a piece of retina from somebody who's recently deceased and put that into somebody else's eye, and would it actually hook up and work? And the answer to that was no, it didn't work. It was way too late in life. It didn't have the ability, the plasticity to be able to hook back up. What about non-humans? I mean, we put pig hearts in sometimes, so what about using a different species? The incompatibilities is a real problem, so that the handshake that's necessary between a human and another species isn't possible. So that was tried too, but not successful. And then the last one was, what if we can just make them in a lab? And in the 90s, that was just a pipe dream. There were people thinking about that, but really there was no way to do that. And that was until the late 1990s, 1997, 1998, when Jamie Thompson at the University of Wisconsin was the cover of Time magazine for the discovery of embryonic stem cells. And the promise of embryonic stem cells at that time was that they were an unlimited supply of cells that at least had the theoretical potential to make any cell type in your body. So they had that intrinsic ability. Now that was true, but at the same time, how do you get these cells that you grow in a dish to become pancreas or retina or motor neurons from your spinal cord and not make everything under the sun? So the problem in the beginning was that, yes, they made all these different cell types, but they made them all in one big soup. And so there was no way to pull out what you needed, and nobody wanted to put a beating heart cell into your eye. So early on during the technology, it wasn't could they do it, is how do you harness it? It's almost too powerful and too wild. And this gets me to a little bit of a side shoot in my talk, which is the term stem cells, which people like to throw around all the time. It's actually not a new term at all. So stem cells we've known about them for hundreds of years, because we know we all have to come from a finite number of cells. And so there has to be something there beforehand that has the capability of making all the different tissues in your body. And as I mentioned before, we know there are stem cells in our bone marrow that continually make blood cells and white blood cells throughout our entire life. Those are called tissue restricted. So for instance, a bone marrow stem cell can only make blood cells. So a bone marrow stem cell can't make an eye, can't make a retinal cell, can't make a pancreas cell. Unfortunately, there are a lot of folks who will take bone marrow stem cells and just say, I'm going to take a stem cell out of your hip and I'm going to put it in your arm or in your eye or in your brain and try to confuse you by just saying it's a stem cell, when in fact it only has the potential to make other types of blood cells. And that's why one of the comments here was on the bone marrow. It's a specific type of tissue-restricted stem cell. The type that we're talking about are called pluripotent stem cells. So that's the key. The term is pluripotent. Pluripotent means it has the ability to make all the different cell types in your body, not just one tissue type. So what is a stem cell? So we'll get into the definition of the stem cell then. So it really is quite, for a pluripotent stem cell at least, it's not that hard to define. So a pluripotent stem cell has to have the ability to divide and make more of itself indefinitely. So one of the great things about stem cells is that by definition, once you have them, you can make as many of them as you want, forever and ever and ever. So there's not a limit to how many times they will divide and make more of them. So you can continue to propagate them in the laboratory. And then if you touch a button, so I say that facetiously, but if you do the right things, you add the right cues, the right chemicals, the right environment, then you can get them to differentiate or become not just a stem cell. I'm going to a little bit of a cold 2 weeks ago, but I'm getting over it. But if you push them along the way to differentiate or develop, they'll then create mature cells. And so those different mature cells can be, again, theoretically anything in your entire body. What my job was to get them to say, I want to be retina, and I want to be photoreceptors, and I want to be retinal pigment epithelial cells. So getting back to the sources. So back in the late 90s, the only type that we had were the embryonic stem cells. And if you recall back at that time, very controversial. They were obtained from from in vitro fertilization clinics from other embryos that would otherwise be discarded. And there were a finite number of them, and they were the cell mass, or the cells that inside of them, were taken, and Dr. Thompson found a way to grow them in a dish. And what he showed was that they were able to produce all these different cell types. Then Dr. Thompson, as well as Dr. Yamanaka in Japan, around the exact same time, found another way to make the same types of cells in reverse. So you could take a skin cell or a blood cell from any living human being, and then through genetic mechanisms, convert them back to a state that was like an embryonic stem cell. And those are called induced pluripotent stem cells. So there's two types of pluripotent stem cells. There are embryonic stem cells, and there are iPS cells, or induced pluripotent stem cells. The iPS cells can be, we've made literally hundreds of them, and any person walking the face of the earth can donate a little piece of skin or a blood sample like they might give for a cholesterol check, and we can produce the same types of stem cells and do essentially the same things with them. And they look the same in a dish. Those are those little colonies. They look just like geometric shapes, but every one of those is thousands of different stem cells, all of which that have that capability to make all the different cell types in your body. So that's a lot of discussion on just where we start with the kind of the clay that we use. And then from there, the trick is how do we get it to make retina? And so that's what my lab started to work on in the mid-2000s. And it took us a number of years to figure out how to do that. And how we learned how to do it was essentially taking the principles of biology that we already knew about how the eyes and the retinas grow from the brain during development. So we already knew, studying different types of species, how the eye develops. It develops as a couple of little outpouchings while the brain is developing. It pouches out, and then over time it envelops on itself. And we know the kind of chemicals that are important for that process and what kicks them into going into the different cell types. And so the key was to be able to apply that knowledge to the stem cell technology that we had developed, and to see if we could recapitulate that in a dish. And so this is in a 3D view, kind of what it looks like. As the eye starts to outpouch from the developing brain, it then kind of folds in on itself, sort of like a pot. And then that ultimately gets enveloped by the white part of the eye, called the sclera, and the lens and the cornea all gets made in front of it. And so the steps in this is a tremendous oversimplification, but what this flow chart is showing is all the different forks in the road, all the different decisions that those cells have to make to determine what they want to be ultimately when they grow up. So you first have to decide what lineage of cell you want to be, then you have to decide you want to be a part of the brain, then you have to decide do you want to be a part of the top part of the brain, not the spinal cord, and then you have to decide you want to be retina and not your cortex. So all those different steps have cues to them. And so understanding what those cues are help us to be able to usher these cells step by step from this very clay-like material, the pluripotent stem cell, all the way to something as specialized as a photoreceptor cell that can detect light. So these are the different things that we use. Timing is important. Cues or chemicals are important. Physical forces are important. And ultimately, this is what you get. So we developed this, and what we found after many, many years is our ability to make these what we call golden Cheerios. So each one of these is like a little cup, like I showed you earlier in a cartoon form. And every one of these is about the size of a large pinhead, so maybe 2 to 3 millimeters. And we can make literally thousands of them. We can now do them in 3-liter bioreactors, so giant, like 2-liter jugs of things. So we can make quite a few of them. How do we know that they're retina? So over time, we see that they have, it's hard to see here, but they have like a fuzz to the front of them, on the top of them. And when we first saw that, we were very excited because we thought, well, could that fuzz be those antennae, those light-catching antennae I showed you earlier? The rods and the cones have those very specialized little proboscis that are designed to detect light. And of course, that's the business end of a photoreceptor. So you can make something in a dish, but if it can't do the job, it's really just a parlor trick. So what we found was, and this is a cutting through one. So if you cut through one of those, you see it's kind of hollow. So it's kind of that bowl shape to it. So we're just looking at one plane. All of the red cells on the top part are cones. So that's a marker that turns it red if it's a cone cell. Those cells that detect color and high detailed images. The purple cells below that are all the rods. And then the green cells below that are cells that haven't decided what they want to be yet. And if you look at this compared, and this is an actual retina from a monkey, it looks not exactly the same, but pretty similar. So for something that started out as a blood cell from a person from Milwaukee, it's pretty remarkable that we can make something as similar to, as it is to a non-human primate retina. And so this is where I'm going to walk over and get this going a little bit. So if you look at the surface of one of these, oops, let's go back. There we go. So if you look at the surface of one of these little golden Cheerios, the red dots and the green dots are all cones and rods, and they're all dotting the surface. And you get the sense that they're these hair-like, little, like it's got a fuzz on the top of it. So those are all the light-catching, what we call outer segments, those antennae. Now if we zoom in then, so that's a pretty low magnification image. Now if we zoom in on where those, walk away and it doesn't behave. Okay, so if we zoom in on that, you can see again, it has that layer to it. So all those red cells are cones, and the top part of that is where it's catching light. Those are all the kind of the hair-like portions of it. And then it has those long lines that end in what look like feet. And that's where it's making connections with the next cell type, the bipolar cell. So not only is this a retina that has the photoreceptors in it, but it has all the different layers. So it will detect light and pass a signal all the way through it. Now, of course, it doesn't go anywhere because there's no brain attached to it. But it has all those parts, all those thousands of little proteins that I showed you earlier that need to be there to make it an authentic photoreceptor. They're all there. What I'm showing you here in yellow are the rods. Most of the red ones were cones. Oh, I think we're good. Okay, so what I haven't talked to you about is the retinal pigment epithelium. So one thing that we can't do is grow them together, so as of yet at least, but we can take those same golden Cheerios, and if we look at how they grow, once we pop them off in the laboratory dish, what they leave behind is a skirt or like a carpet around it. And if we treat those a certain way, we can get them over time to produce the retinal pigment epithelium. So we can make both of those essential parts. We can make the photoreceptors and we make the RPE. And these are authentic spare parts that are the same as what you have, close to the same to what you have in your own retinas. And so that was the first part, the manufacturing. Can you manufacture authentic cell parts for your retina? And the answer to that is yes, and that's what we and others are doing right now in large quantities to put forth in front of the FDA for approval for patient therapies. But that's not where it ends. So you can make a piston, but can you install a piston, right? So a piston laying on your driveway is not going to do you any good, right? It looks good. Now, I should say, I don't want to be too flippant about that, because even what we do right now, even if we don't put this into a single patient, what it allows us to do is it allows us to understand how cells die in disease. So if we take one of these, if we take a blood sample from a patient or a child or an adult that has a particular disease, we can make their retinas in a dish, and we can study them. Now, we can't do that in an actual human because we can't take it out of their own eyes. But now we can make them an unlimited supply, and then we can test drugs, we can test gene therapies, we can test things in the laboratory on their retinas to see whether or not they work. So it's very, very exciting and beneficial to patients, even if it doesn't go back into their own eyes. But of course, that's ultimately kind of what a lot of us want to do. And so kind of in the top there, all the little yellow dots you see there, are all those golden Cheerios, just thousands of them. You can vial them. And this was what was the foundation for the company in Madison called Opsis Therapeutics that was spun out from the university. And so this is one of those 3-liter bioreactors. Let me get this going. And so what you see spinning in there are, again, this is over 60 billion photoreceptors that are being created in one of these 3-liter bioreactors. And this allows you to start thinking about treating large populations. So there's enough in this tandem 3-liter setup to treat a city the size of Corpus Christi, Texas, which is really quite remarkable. We're talking about 10 years worth of 10 to 15 years worth of research from starting with just the clay. So I mentioned before, installation's a big deal. So if your purpose is not to just study the disease and to develop therapies in a dish, which are very helpful to patients, but at earlier stages of their disease, because again, once your cells are gone, a drug isn't going to help you, a gene therapy isn't going to help you, because that's like showing up at your stoop with a new furnace and the house is all burned down, right? So if the house is all burned down, you need a new house. You don't just need a new furnace. So how do we do that? So pretty crude at first. The idea is like, okay, well, what if we just took it and kind of put them back there at the right place, and maybe they'll find their right way. Maybe these are plastic cells, and maybe they'll find the connections that they need to find. And so the early steps were something, keep it simple. So don't make something more complicated than it needs to be. So there are very straightforward ways in which you can insert a needle into the subretinal space in a patient or an animal model, if you're using an animal model, and just inject them in the area where they used to be and see what happens. However, that actually has a lot of promise to it. It has a ceiling, so it may be able to help people a little bit, but it probably has a ceiling because, as I showed you before, the cells aren't just randomly positioned back there. They have a very precise orientation, very precise architecture. So is there a way to utilize the tremendous amount of brain power and resources that we have at the University of Wisconsin to try to take those cells and to arrange them such that they may have that proper orientation? So we turned to Dr. Gong and Dr. Ma, two wonderful engineers. Dr. Ma actually works on making computer chips. So he's very used to making very tiny, tiny little architectural features in very small areas. Never worked on anything biologic before. And I said, can you do what you do on a computer chip, but do it for something I can put a cell into? He loved that idea. And it turns out that his spouse, Dr. Gong, who's a world-class materials engineer, knew what types of materials were necessary that could biodegrade and were compatible in such a delicate area as the back of your eye. So we combined all of our kind of expertise together to make these very thin film little scaffolds. So the original design that we had looked like a wine glass, and so we could put an individual cell into each one of the wine glasses. And when we did that, we could align them. So they were oriented all correctly. The problem with this design, which we loved a lot, but it had a lot of biomaterial, it had a lot of things that had to dissolve in the back of your eye, and not as many cells as we need. We really need to pack them in there. So when we mentioned that to Dr. Gong and Dr. Ma, they said, well, let's change the design a little bit. So we went to this kind of ice cube tray design. This was recently in a lot of the news outlets that we just recently published, where we can seed those same cells, we can pull them out of those golden Cheerios, and then pack them into these ice cube tray designs. And they're very flexible, they dissolve, and we can get them into the back of the eye. And this is an example. So we worked with the National Eye Institute in Bethesda to be able to devise a way. And you guys hopefully had dinner a while ago, so it'll be all right. So this is a pig eye. And so what we're doing here is making a small hole under the retina and then making a blister. And this raises the retina up. We then make a small incision in the retina. And that allows us to, and it'll be here in a moment, to place the scaffold with the photoreceptors in them. And here's this is a great part. Disclaimer here. This is what we do. It doesn't hurt. And this is the injection device. You can't see the scaffold. It's kind of outlined in green there. You can't see the scaffold because it's transparent. And then it's being injected now into the subretinal space where those green arrows are. And this is in a pig model. This is a pig model that also has blindness to it. So now we're actively working. We actually have a Department of Defense grant that we're working with. So we're working with the Department of Defense to do this not only for folks that have degenerative diseases of the eye, but also soldiers who have combat injuries so that they have blast injuries or laser injuries to their retina. So we're expanding our work not just for the kids and folks with AMD, but also soldiers who lose sight through injury. So once you do that, yeah.
A live pig?
That was definitely a live pig. Yep.
Could he
Yep.
tell the difference? You said he didn't have the color or couldn't see before?
So these pigs have a natural degenerative disease. So they lose their ability to see. Pigs don't need to see very much. They use their nose quite a bit. But there's facilities that you could do. Is the ORs look just like human ORs. Problem with a pig is they're generally a lot bigger than humans. So you have to. It's a little bit of a delicate process, but the pig is oftentimes used as a surrogate for human because the eyeball is almost exactly the same size as a human's eye. This is a color image of a pig eye that received one of our scaffolds. So the pig portion of it is all the green that you see. But that pig doesn't have any photoreceptors. So what you see with the red dots, every red dot is a human cell that has been placed into that same spot where the photoreceptors used to be. And at this point, we're testing these animals to see if they restore vision, but it's still human into pig. And so the problem comes in with the fact that the different species really don't shake hands with one another. They have different proteins that don't interact. So what we're doing in addition is we've made pig iPS cells. So we can make stem cells from pigs to then make the photoreceptors to go back into the pigs. And so this is the kind of quilt you have to put together when you approach the FDA, because you get to these impasses that just, it's not going to work. It's a catch-22. So you have to kind of think through it and say, well, how can I convince somebody that it might work? You have to go into the same species. But you're not going to put a pig stem cell into a human, so you have to do them in parallel. So in the end, you get kind of this relatively complex 8,000-page document that kind of tries to convince people that it's safe. That's the most important thing, safe, safe, safe. But then also has a chance of helping patients too. Okay, so who do we use? So I told you before, we can take these stem cells from any human being walking the face of the earth. The problem is to make those stem cells and to differentiate them into photoreceptors, put them into all the things we need to do. And if we did that on an individual basis, right now it would be multi-million dollars per person. So it's really not a sustainable, economically sustainable thing. If you're Jeff Bezos or something like that, maybe, but not for the regular stiffs like us. So you really have to think of a way, how can we use something that is more translatable to large populations so that we're not a huge burden on the healthcare system. So personalized, yes, you can do that. If there's a genetic defect, you also have to fix that genetic defect, or you're just putting another disease cell back into the same patient. So it can be done, but it's expensive. Is there a one-size-fits-all? That's kind of what we're looking at. We're trying to develop a process whereby one cell type, one photoreceptor cell type, one RPE cell type off the shelf can go into every individual. The downside of that is it's probably going to require some drugs, some immunosuppression, some light chemotherapy to prevent you from rejecting those cells because they're not exactly yours. So there's a trade-off whenever you go with something that's more generally applicable, and that is the medications that you have to take to keep those cells alive may have side effects too. And then there's this idea of, can we partially match them? Can we have a bank of multiple different types from different individuals whose background is similar, sort of like matching a kidney to you? It's not perfectly you, but there are parts of it that are more like you than the person next to you. That's also something that's possible and that we're looking into, but is expensive. Okay, so all well and good, but all of this, is it going to ever reach patients? Well, the answer is it already has. So the retinal pigment epithelium, just the pigmented part, the cobblestone brown and blackish cells, those have already been put into dozens of patients in different trials. The problem is that they don't detect light, right? So they're put in, and the idea is that hopefully they will revive some of the photoreceptors that are already still there that aren't quite dead yet, so that they can hopefully improve somebody's vision. It is shown to be safe thus far, but the vision improvement has been modest or non-existent at this point. But that's a little bit expected because, again, the people that are taking them have lost photoreceptors, and they're only really getting the helper cell and not the cell that's doing the job of detecting light, because we haven't been able to make the cells that can detect light in large batches like we can now. And the National Eye Institute, I mentioned earlier we were working with them, Dr. Kapil Bharti, they have this entire initiative called the Audacious Goals Initiative. And that initiative is to prompt scientists and clinicians around the nation to look in, sort of like, you think about making a vaccine, right? If you put enough effort into it, you get people to work together and hopefully you can do something big. And so they recognize this as one of their audacious goals, and that is to replace photoreceptors. So they certainly believe in it. And so do we, but you have to be smart about it and you have to be honest and transparent. It is not as fast as we want it to be. It has to go at a certain pace that we hope is as fast as it can be, but it has to be safe, and we have to make sure that we're doing the right thing and not cutting corners that can ultimately hurt a patient. And so that does take time, but we at the McPherson Eye Research Institute have a large group of folks that are specifically dedicated to restoring vision using stem cells and other techniques. And of course, Diopsis Therapeutics was started as well. And so there are industry efforts, not just in Madison, but now there are probably a dozen or more companies around the world that are looking to do the same thing, which is cell replacement in the back of the eye. And they're putting hundreds of millions of dollars into this. So clearly we've gotten to the point where federal governments, industry are taking notice and feeling like this is going in the right direction. These are some of the trials. I mentioned that there's a lot going on right now. These are trials that are either ongoing or are planned in the near future. So there's quite a few of them. And kind of most importantly, what I always like to end up with in all my talks is the managing expectation part. So what are we hoping for? Are we hoping to take folks who have lost all of their ability to see and erase the vestiges of a devastating disease with one injection? That's unrealistic, right? So that's like saying to somebody who's a quadriplegic, well, we're going to put a stem cell in you and you're going to run a marathon. So really it's steps, right? We didn't get computers, supercomputers on our wrist overnight. Initially they were machines that could add things and they took up a whole room. And then over years, because they showed benefit, people innovated and they got better and better over time. So airplanes, automobiles, computers, there's this process for all these technologies. And the same thing will happen in biology and therapeutics and stem cells. So it's unrealistic to expect a miracle out of the gate. It's not going to be a miracle cure, but I think it's going to have a role. And that role, if it shows benefits, will continually to get better over time and hopefully provide people with better and better vision and more options. But we do have to crawl before we walk, before we run. And until we get to a point where we really can help people, there are unscrupulous folks who will try to fill that void with fancy terms and try to convince you to do things that are not healthy for your finances or for your personal health. So what I like to tell folks, and we have a website at the McPherson Eye Research Institute, which is like the top 10 things to watch for before you spend any money on a stem cell treatment. And the number one thing is, if you have to spend money on it, it's not legitimate. So all stem cell therapies right now that are legitimate are in a stage where they're experimental, and they pay you to be enrolled in trials. So if you're paying for something, it's a conflict of interest, and it is almost assuredly snake oil. So places that charge you for anything that refers to it as a stem cell, run away. Okay, so summarize. Technology to build these retinas, it's amazing. To this day, if I wake up in the morning, the idea that you can take a sample of blood from somebody and make these retinas that can detect light and transmit signals is really quite remarkable. But installing them is still a significant challenge and one that we're working on, and that's where I think most of the innovation is going to have to go in the foreseeable future. And all of this is, thankfully, and I'm very proud of this, very uniquely situated in Madison. So now we have almost every institution in the world has a stem cell center now, because it's been recognized as being such a powerful tool, not only for therapeutics, but for just understanding how different tissues are developed and how to design drugs and gene therapies as well. But almost all of that had its roots in Madison, Wisconsin, which is really something for us to be proud of. And then, like I said before, I get to talk about all this, but I have a lot of folks over the course of the last 15 to 20 years that have passed through my lab that are brilliant, that come up with wonderful ideas, that actually come in super early, stay super late, and they don't get enough credit. This is just a snapshot from a few years ago. Some of the players have changed, but just want to let you know there's a cast of tremendous characters behind all the work that I've shown you today. Also, there's a lot of support that goes into this, federal support, both through the National Institutes of Health, the Department of Defense, and then a lot of different foundations. The McPherson Eye Research Institute itself, and folks that contribute from Wisconsin and around the world that we're very grateful for. And these are some of the collaborators that I mentioned during the course of my talk today, other professors and scientists and clinicians that have taken an interest and helped to really advance this research over the years. And my lab is just one of over 60 to 80 at the McPherson Eye Research Institute. So everything I've talked to you about today is just my lab. So you multiply that by 80 to 100, and you get the idea of what's going on at the University of Wisconsin in Madison. So it's really quite something. And as directing the Eye Institute is quite a privilege because it allows me to get to know these other folks, all these other dots on the wall here, and to be able to develop collaborations and projects with them to advance everyone's research and hopefully help patients, which is what we were created to do. So thank you for your time, and hopefully it was not too painful to have to listen to all that, and I'm happy to answer any questions.
Thank you very much. Before we get going with the Q&A here in the room, I wanted to repeat the telephone number for our viewing audience, 715-942-9917. I think we've got a question right here.
Yeah.
I understand the cooperation that you've been getting from the U.S. and even outside of the U.S., but are there other areas where you don't receive that much cooperation, and therefore there are people who are working on some of the same ideas you are, but they're not willing to share the information?
I wouldn't say I have not in my, maybe I'm Midwestern too much, but in my experience, I have never bumped into anybody that I thought was antagonistic or kind of siloed. I mentioned this a little bit earlier. It's Wisconsin is fairly unique in that I have never met anybody on campus who wasn't absolutely happy to have a cup of coffee and tell you what they did, mainly because we're all conceited. But
I'm thinking more internationally.
Yeah,
You're thinking of North Korea, for example.
Well, I'm not doing anything with North Korea.
No, I'm sure
I'm pretty sure they've cured blindness. So we're just all catching up to North Korea. But yeah, so yes, the world's a big place, and just like with anything else, any other technology, it's good to have competition, right? And so you want to have folks in different areas all trying to do the same thing. Mm-hmm. If there was only one group doing it, who knows that they've got all the right ideas? I guess the point I was trying to make is the vast majority of folks that I've met in science and medicine are happy to work with you to exchange ideas. But you don't work with everybody. So you find out, that there's folks in California doing what you're doing too. Well, God bless them. That's a great thing. I'm glad, and they may reach out to me or not. I can't work with everybody. They can't work with everybody.
I'm just thinking there are so many pieces to the puzzle.
Yeah.
And somebody has a piece somewhere that you need.
Yeah. So that's where publications come into play,
oh,
right?
Sure.
So that's where the whole the global scientific system, if it works well, that's where it comes out, because that's why you publish. Mm-hmm. And that's the balance between industry and academia. That's why academia is so important, because if I'm an industry, if I'm a CSO for a company, and I try something and it doesn't work, I'm not telling you that it didn't work, because I want my competing companies to do the same thing for the next 20 years so I can have a leg up on you, right? Academia is totally different. You publish that. Your goal is to make sure everybody knows what you're doing. You publish it, you get credit for it, right? And that's how you get promoted and notoriety and what have you. And that's worldwide. And so the sharing of the information is integral to academia. And that's why the university systems are so very important. Back in the day, the major companies had research institutes too, and they published as much as everyone else, but that's not the way it is anymore.
I'm sure you have the thousands of people throughout the world that are working on other areas, hair,
Oh, yeah.
hearing, and you name it.
Yep, and we talk to each other too. So there are different groups. So there's a lot of different bubbles I work in. One is vision research. So I work with folks who don't do stem cells, but they're interested in gene therapy or drugs or other things, but they know the eye really well. I also work in stem cell circles, because you can learn something from somebody trying to replace a motor neuron in a person who's paralyzed, or replacing a dopaminergic neuron in somebody who has Parkinson's disease. How did they get them to connect? It's not in the eye, but I can learn principles from them and apply them to the eye. So being able to bounce those ideas or interact with different spheres is very important, just like what I talked about with my biology with somebody else's engineering. And that's really what I've seen in the last 20 years change, is that with our ability to reach out to different institutions, back when I was training, your sphere was really who was in your building. And so if somebody, you had access to the folks in your building, but even your whole campus you may not have had access to, much less somebody else in Wisconsin or a different country. Now it's a much smaller world, and people are more apt to try to take what they know and add it to somebody else's, and that now is considered something that's very admirable. 20 years ago, you really had to be independent, right? You were rewarded for being a lone wolf, and thankfully that attitude has changed.
Here's a question online from Richard Hansen. What can be done to slow the progression of AMD? And secondly, how effective are specific eye vitamins currently available over the counter?
Yeah, so first and foremost, you don't smoke. So by far, smoking is the largest risk factor for progression of AMD. So simple lifestyle changes are the most important. A lot of AMD, unfortunately, is genetics, so it tends to run in families. That you can't change, at least not right now. The vitamins, you get a lot. There's a lot. Ocuvite, Preserve Vision, they get a lot of attention on in advertisements and things like that. But if you look at the actual studies performed with them, they helped to slow down significantly, but not very. The statistics were correct, so it does work. But there's a particular type of folks with AMD that have these kind of what we call moderate drusen, and if you looked at that group only, their rate of vision loss was slightly less than folks that didn't have them. It did not prevent, so if you took it ahead of time, it didn't prevent you from getting it. If you had severe, it didn't slow it down. If you had mild, it didn't slow it down. So it's being used kind of as a panacea because there's nothing else you can do. But honestly, what I tell folks, and they're expensive, so I tell folks, you don't smoke, you get out and you walk, you exercise, and you eat a balanced diet with green leafy vegetables and things like that, and you're doing as well as you're doing with the vitamins too. Doesn't sound like very much, but it oftentimes with folks with fixed incomes, when they look at the price of some of these vitamins, it helps from a financial standpoint. But beyond that, one of the reasons we're doing what we're doing is there's not much more you can do.
Any more questions in the audience?
Yeah.
Is this one?
Yep.
Yes.
Thank you for that wonderful explanation
Well,
of
that's-
stem
well, thank
cell, because it sounds so mysterious and how it helps. I appreciate that. Thank you.
Oh, well, thank you very much. Yeah, it's a lot easier to say, here's a stem cell, it'll cure what, it's hilarious. You get some of these advertisements that you see, and do you have eczema? Do you have, do your joints ache? Are you deaf? Or you have, rub this on your forehead. And perfectly smart people, that would never buy a Brooklyn Bridge, because they're desperate, will fall for these things. And frankly, I think it should be criminal, and unfortunately it's not. Oftentimes they pass it as a nutraceutical, which means it's totally unregulated by the FDA, and so it's not illegal. But then again, and there have been cases like in Florida, go figure, Florida, that where folks have taken, here's the most egregious example I can think of, and that is where, you have fat stem cells too, by the way. Go figure, right? But they're stem cells. And so people have actually taken, it's a twofer. You get a liposuction, so that's good, right? And then they take the stem cells out of your fat and they stick it in your eye. right? And they blinded people doing that, right? But people paid $40,000 to $50,000 to have that done because they were convinced that the stem cells, which are just mysterious stem cells, was going to cure their blindness, right? And there was nothing illegal about doing that because it was autologous. So because it came out of the patient and went right back into the patient without being manipulated, it's a civil lawsuit if you want to, but it's not a criminal lawsuit. So those things happen. They happen. It used to be you had to go to Ecuador to get that kind of messed up sort of thing, but you can do it right here on our own country's shores now. So part of why I do this is not only to educate, which thank you for saying that, but also to steer people clear of things that aren't ready for prime time. Bob.
Dr. Gamm, can you talk just a little bit about some of the clinical trials that are currently going on, and when do you think we might have a significant clinical trial for AMD?
Well, all of the ones that have been done thus far, almost all of them have been for AMD. But they've been for that helper cell, the retinal pigment epithelium. There's a reason for that, and that is that cell type is actually one of the easier ones to grow from stem cells. So way back in the mid-2000s, because it's pigmented, it's brown, you can find it. You can find it amidst all the other stuff, and you can pick it out, and you can grow it separately. So there were just logistical reasons why it was one of the simpler ones, more straightforward cells to replace in the entire body. So the first one that was tried was a motor neuron, which was a bad choice because you had to stick it in the spinal cord. And I mean, how is anything going to get from my spinal cord to my toe, right? I mean, that's a pretty big leap to start with. But the second cell type that was tried was the retinal pigment epithelium. And they went for macular degeneration because it's a cash cow. There's millions of people that do it. So if it works, then you've got, the industry was really interested in that. But from a conceptual standpoint, didn't make a whole lot of sense because they were putting it into patients that had both the photoreceptors and the RPE gone, and they're just replacing the photoreceptors. Now, I don't malign it too much though, because there is the possibility that the photoreceptors from the patient that may not be working but still aren't dead yet on the edges of their blind spot might be able to wake up and then shrink their blind spot a little bit. And anybody that has AMD will tell you that they'll tell you as soon as their blind spot gets a little bit bigger, because it really does affect their vision. So that's a meaningful change. If you can kind of shrink somebody's central blind spot even a little bit, that's a reasonable endpoint for a study like that. And in fact, they did find that in a few of these early phase 1 trials where they're only really treating about 12 to maybe 18 patients. What they also showed was that these cells, which were, for the most part, not from the individual, so they were a one-size-fits-all, that they were well tolerated. So they didn't cause people to lose additional vision. There were some surgical complications that were parts of some of the surgeries, but the cells themselves didn't seem to cause harm, which is always an important part in these early studies, because there's always something to lose. And so no matter what our good intentions might be, people have things to lose, and we want to make sure that it's first and foremost safe. So all of these early trials were aimed at AMD and pretty late stage AMD. They were safe, showed maybe in a few patients a little bit of improvement, nothing to write home about, but it really served as an important stepping stone for the next stages, which is to try to replace the photoreceptors in addition to the RPE cells. So like I said, it's an iterative approach to developing these types of complex therapeutics. And just now, there's one patient, I think, in Japan that got not just the photoreceptors, but the photoreceptors mixed in with a lot of other of the bipolar cells and kind of a more full thickness retina that got treated with that within the year. We have no idea how that patient's doing. It hasn't been published yet. But these things are starting to happen. But again, it's not going to be a grand slam home run right off the bat. So I think you have to be ready for the fact that it's going to be an iterative process, but if it's done correctly and safely, then the process will continue. The problem is if somebody, cowboy goes in there and does something they shouldn't, and somebody gets hurt, it'll put the kibosh on it for years. And that's what happened for gene therapy. Gene therapy got ahead of its skis and people didn't really understand it as well as they should have. Some folks got a type of cancer from the early gene therapy trials, and the FDA shut it down for over a decade. Basically said, we're not doing this anymore until you do, all these various things. So, and then it was, ironically enough, reborn because of a gene therapy in the eye for a childhood form of blindness. So the eye came to the rescue on that. But again, we don't want that to happen with stem cells. We don't want to be in such a rush to get to whatever the finish line might be that we actually send ourselves back decades.
So your research and efforts are all directed at finding a way to replace the dead cells. Is there any research or effort being put into finding a way to trigger these cells that don't replicate themselves to replicate themselves?
Yes. So basically, I'd say about half of my lab replaces them. The other half of my lab uses them in the dish to make gene therapies and drug therapies. So I do kind of do the whole gambit. So it's not just cell therapies in my lab. But what you mentioned is what we call a photoreceptor regeneration. So there are laboratories. So interestingly, certain types of salamanders and fish, if you rip out their retinas, they regrow them. Right? Now, they have a very simple retina, and they basically, their food doesn't move. They eat plants and things like that, and they just need to have retinas that detect movement so that they can scurry away. So they have very simple retinas, but they have the ability to regenerate. And so there are laboratories that look at that and say, well, what kind of triggers do they have that allow their neighboring cells to fill in the gaps that humans and other mammals don't have? Because it's not just humans, mammals can't do it. And so people have looked at that, and there is a significant amount of effort going into trying to take some of the cells that are still alive and turn them into photoreceptors. Thus far, they have, there's groups, there's a good friend of mine in Seattle who works on this a lot, that takes a particular type of cell type and has shown that it can divide and form a new type of one of the bipolar cells, or the inner types of cells, not the photoreceptors, but another. So it's possible, and it's called dedifferentiation and transdifferentiation. It's possible to get a cell to revert back to an earlier stage, kick off a daughter cell, and have that daughter cell become something different. But they're, at this point, very crude. They're not necessarily hooked up, and they're not photoreceptors. But that is a very prominent area. So that Audacious Goals initiative that I told you actually has two parts to it. One is what I talked about, the replacement. The other part is what you're talking about, which is the commandeering cells from the actual person to make the new cells, which of course would obviate any immune issues, but brings up a lot of other hurdles as well that are being worked on. Great question.
Well, I think we've come to the end of the program.
well.
Thank you very much.
Thank you. Thank you very much for the invitation.
Outstanding.